Welcome to the second screencast on genetic engineering. This continues to look at some of the techniques used by genetic engineers, and it focuses on polymerase chain reaction (PCR), and then it also looks at issues to do with engineering of plasmids, which we've touched upon in the previous screencast. So, dealing with this slide H in your notes, this one deals with polymerase chain reaction, which you will have seen actually happening in class, but this goes through the theory of what is going on. Okay, so if you actually look at your slide, which you've got a copy of here, so I'll move away from that, but if you can look at your hard copy of that whilst I explain it through on the diagram below. So here I have my DNA, okay, and the whole idea of polymerase chain reaction is that what we're trying to do is amplify the amount of DNA, i.e. multiply the strands of DNA. Quite often we have very small amounts of DNA, and what we want to do is to make sure that we have enough of the DNA so that we can use it for all sorts of things, such as genetic engineering, or perhaps even taking a genetic fingerprint, and also perhaps involved in working out the genome. So one of the things that we're going to be doing is to try and increase the amount of DNA that we have, and this process known as the polymerase chain reaction, or PCR for short, is how we go about doing it. Okay, so what we do is that we have in our mixture, we have the DNA which we're trying to amplify, we have small strands of DNA which are complementary to regions of the DNA that we're trying to amplify. Okay, so there's complementary to bits of the DNA, and these are called primers. Okay, we need two different types of primer, a primer that will bind to one half of the DNA, and a primer that will be complementary to the other half of the DNA too. We also, in our mixture, have nucleotides. Okay, so these structures down here represent free nucleotides. So we need lots and lots of those of our mixture of all the different types of nucleotide: adenine, guanine, cytosine, and thymine. Okay, so we have all four of the different types of nucleotide present in our mixture, and we also have this represented by this red squiggly thing here. We have enzyme DNA polymerase, which you'll remember from module F215 is the enzyme involved in DNA replication. Okay, so this mixture of our DNA, our primers, our free nucleotides, and our DNA polymerase is all put into a special machine called the PCR machine. Just one thing I ought to add to that is the DNA polymerase that we use in here is a special DNA polymerase which is extracted from thermophilic bacteria that we find in hot springs. Okay, so those hot springs you see in countries like Iceland have bacteria that live in them, that live at up to 90 degrees centigrade, and obviously they've evolved proteins that don't denature at those temperatures. Okay, so these thermophilic bacteria have a DNA polymerase, okay, which doesn't denature at the temperatures that DNA polymerase for most species would. Okay, so moving back to my PCR, what we then do, the first thing in our machine, what happens is that the DNA is heated up to 95 degrees centigrade. Okay, and what that does is it separates the two strands. Okay, the heat breaks the hydrogen bonds, holding the two strands together, so we ended up with single-stranded DNA here. Okay, then what we do is that we cool the mixture down at about 60 degrees centigrade. Okay, so the mixture is cooled. Sorry, in my notes I've got 55, so we'll go for 55 degrees centigrade. So it's cooled down to 55 degrees centigrade. And what happens at 55 degrees centigrade is that the primers, which we talked about, they will bind to complementary sequences on each half of our DNA. Okay, and then what happens is once we've got a double-stranded molecule here, what will happen is that the DNA polymerase, okay, will recognise this, okay, because the DNA polymerase wouldn't recognise single-stranded DNA because its active site will only recognise double-stranded DNA. That's why we have to have the primers. So the DNA polymerase binds to this region of double-stranded DNA. And what it does is it will move along the strand of DNA, adding free nucleotides and causing the phosphodiester bonds to create the sugar phosphate backbone, so that we end up with a strand of double-stranded DNA. Okay, so by the end of that process, we've doubled the amount of DNA. So we started with one section of double-stranded DNA, and we end up with two sections of DNA. Okay, and then what we do after we've done that, you can then heat it back up to 95 degrees centigrade, and then cool it down to 55 degrees centigrade, and it will double again. So after we've done that, we end up with 4. Okay, and then you heat it up again, cool it down, you get 8. Okay, and then you can do the same 16, and you can keep doing that. So you can very, very rapidly amplify your original DNA signal. Okay, to having hundreds of thousands of copies of your original strand of DNA. Okay, so that's known as the polymerase chain reaction, and I've got a little animation which will hopefully further illustrate the whole idea of polymerase chain reaction. So if you just follow this through, reading it through as it comes along, PCR stands for the polymerase chain reaction. Okay, and it's a mechanism amplifying the DNA signal. And a simple device looks like that, which I've shown you in the lab. So the tubes were the things that we talked about, are just added, and then it's heated up and cooled down, and every time it gets heated up and cooled down, the signal is amplified times 2. Okay, so just read this through. So moving on to the next slide. This slide, it says, explain how isolated DNA fragments can be placed in plasmids with reference to the role of ligase. Okay, so in the last slide we talked about restriction endonuclease enzymes, or restriction enzymes, which cause staggered cuts at regions, specific base sequences on the DNA, and these staggered cuts result in bits of single-stranded DNA that stick out from the ends of where the restriction enzyme is cut. We call these sticky ends. Okay. Now, if we cut a plasmid with the same restriction enzyme as we've cut a piece of DNA, what we'll get is the loop of the plasmid will be cut open, and each, if you like, end of the loop will have a complementary sticky end to the gene that we've also treated with our restriction enzyme. Okay. And the joy of that is that the sticky ends will be complementary to the sticky ends in the plasmids, and therefore the gene will fit into the plasmid. Okay. However, the story doesn't stop there, because the complementary bases that are formed with the sticky ends will just anneal together without the sugar phosphate backbone being completed. Okay, to complete the sugar phosphate backbone, we need another enzyme called DNA ligase. Okay, so key word there, DNA ligase. Diagrammatically, okay, diagram I think I've shown you before, is we've got our two bits of DNA from different origins. So this might be our plasmid DNA, this might be our DNA from our gene. They've got complementary sticky ends here, TTAA, okay, and AATT. Okay, they will bind together. Okay, but to be able to knit the two together in terms of finishing off the sugar phosphate backbones, we need this enzyme DNA ligase. Okay, and showing that again. So here we have, okay, our plasmid. We've cut this plasmid, our staggered cut. Okay, with our sticky ends. We've also cut our gene, which has got sticky ends at each end. We then insert that into there, but to knit the whole thing together to make the sugar phosphate backbones, we need this enzyme called DNA ligase, which completes the insertion of this gene into this plasmid. There's a nice little animation of that, which I'll just let you watch because it's quite fun. So this is DNA. This is a restriction enzyme which is cutting it open. It's looking for its base sequences, which it's going to cut at. And it's moving along until it finds the relevant base. It's found the base and it's cut, and you can see these sticky ends. Along comes the gene. The gene finds its sticky ends. Complementary base pairing is occurring. And then along comes the DNA ligase. So here's the DNA ligase, which is knitting it all together again. The same happens at the other end. So moving on to... Okay, so we talk about plasmids a lot, but plasmids aren't the only things that are involved in as acting as vectors, which vectors are things that will be able to get our target gene into our host cell. I say plasmids are often used, but there are other things as well, and we'll have a look at a few of these things. Okay, so other things can be used as well. So the virus DNA, I think I've mentioned before briefly. I'll look at that again in a little bit more detail. Okay, viruses, the life cycle of viruses. The virus is very simple. They're DNA and a protein coat, and what they do is they inject their DNA into cells, and the DNA then makes more protein coats, and then those protein coats burst out of the cell and infect other cells, and that's how viruses actually work. So the whole business of injecting DNA into cells is what they do. So that's a useful way of getting DNA into cells for genetic engineers. We'll look at that. Some bacteria could also invade cells and get their DNA into cells that way. And when we do our golden rice case study, that's the way that we get DNA into golden rice. We'll explain that later. And then see things called liposomes. Okay, we'll look at what a liposome is in a second. So here we go. Here, first of all, here's viruses as a vector. Virus as a vector here. So here's our virus. Okay, the virus has got its own viral DNA, and it's got our gene that we've inserted in here. So we would have inserted that in here with restriction enzymes, sticky ends, ligase, etc., and then got the DNA into the virus. Okay, and then in this case, the virus actually invades the cell entirely. Okay, but then what happens is that the virus sticks onto the wall of the nucleus of this cell and actually injects. You can see here it's injecting its DNA directly into the nucleus. Okay, so this is the virus is the structure which is injecting the DNA into the nucleus. Okay, some viruses don't actually enter the cell at all, they just inject their DNA directly into the cell. Okay, particularly viruses which attack bacteria, because as you know, bacteria don't have nucleuses, so the virus will just inject its DNA directly into a bacterial cell. Okay, and the second type of vector which I mentioned there, okay, is a thing called a liposome. Okay, a liposome, here we go. And a liposome is very simply it's like a vesicle. Okay, it's like a small vesicle, okay, with a phospholipid bilayer, okay, forming the spherical structure, okay, and inside that spherical structure we can have DNA, okay. I've got this diagram, I don't know where I got this diagram from, but you can also notice that it can also be used as a vehicle to get drugs into cells too. It's not relevant for what we're looking at here. We're interested in DNA in this particular case. Okay, and what a liposome does is it fuses with the cell surface membrane, okay, because this is phospholipid, so it can fuse in the same way that lysosomes and vesicles fuse with membranes. It fuses in exactly the same way, and as what happens is therefore the DNA is then released into the cell cytoplasm. Okay, so it's like exocytosis in reverse, or perhaps endocytosis is similar to, in terms of this being a lysosome. Okay, but it's more like the opposite of exocytosis. Okay, so you can imagine exocytosis where a vesicle fuses with a cell membrane to release its contents to the outside. This is doing the other way. The liposome is actually fusing with the outside of the cell surface membrane and releasing its contents into the cell. Okay, so that's how a liposome works. Okay, moving on to the next. Okay, I've also mentioned in my previous screencast, I mentioned a bit of this before, but here's another method. It says, explain how plasmids may be taken up by bacterial cells in order to produce a transgenic microorganism. A transgenic microorganism is a microorganism which has had a gene inserted into its DNA, a transgenic microorganism. Okay, and as that can express the design. So with the idea, this could be our insulin, for example, is actually how do we get the plasmid into the bacteria so that that bacteria can express that gene and produce our insulin. Okay, so we talked about in the previous screencast, I talked about electroporation, I talked about microinjection, and I also talked about the use of viruses. Okay, here's another way of doing it, okay, which involves sort of heat shock treatment. Okay, so what happens is we rapidly cool the bacteria in the plasmids, okay, to freezing point, okay, and then very rapidly we heat those up to 40 degrees centigrade. Okay, and what happens is this seems to result in an increased uptake of plasmids by the bacteria. Okay, so it increases the rate at which bacteria will take up plasmids. Okay, and therefore have a much higher success rate of transforming the bacteria. Okay, we talk about bacteria that has taken up a piece of DNA, a piece of foreign DNA, has been transformed. Okay, and once they're transformed, the bacteria can then express that gene to produce, let's say, insulin. Okay, so just again, as always, showing that on a diagram. As I've got here, we've got some E. coli here, okay, and what we do is that we add some calcium chloride solution, okay, but then what we do, don't worry too much about the calcium chloride solution, but the key thing here is we actually rapidly chill these, so we put them on ice and they actually get right down to 0 degrees centigrade, okay, and then what we do is that we then take our cooled bacteria and our plasmids, okay, and we heat them up very rapidly up to 40, well here it says 42 degrees centigrade, 40 degrees centigrade will do, okay, and then what we do is we plate them onto a Petri dish, okay, and the idea is that these all have a much higher success rate of the actual plasmids being absorbed into or taken into the bacterial cell, i.e. a much greater success rate of transforming the bacteria. Okay, so as well as electroporation, microinjection, viral transfer, we've also got this heat shock treatment that you should be aware of. Okay, so moving on to the next slide. Okay, this is in the syllabus. This slide doesn't necessarily fit perfectly well with this particular piece of the syllabus because it's nothing really to do with genetic engineering. But what it does is it explains why, a bit about what plasmids, the function of plasmids actually in bacteria are. They're obviously very useful to genetic engineers, okay, which obviously wasn't a reason why bacteria have evolved to have plasmids, okay. Bacteria have plasmids for very good reasons, okay, and what they are is that they are a mechanism by which bacteria can share within colonies, genes. Okay, so a useful gene, okay, in one bacteria, okay, the plasmid can be replicated using DNA polymerase to form multiple copies of that plasmid, and that plasmid can then pass to neighbouring cells, which is quite interesting because it sort of almost confronts our view of prokaryotes as being single-celled organisms, because they actually share genetic information with each other in a way that eukaryotic cells don't. Okay, so it sort of challenges our rather simplistic view of prokaryotes being single-celled organisms. In actual fact, there's an awful lot of coordination between the cells of bacteria living in a colony. Okay, and so what happens is that so if you can imagine here's a bacterial cell here with its main DNA, and here is a plasmid. Okay, now what can happen is this plasmid can replicate. Okay, and one of these, one of the copies, passes through a structure which extends between two neighbouring bacteria called a conjugation tube. Okay, so the plasmid will pass through, okay, and will form in the neighbouring bacterial cell. Okay, so this bacterial cell then has the plasmid. Okay, in this case, this has obviously contained a gene which is formed in making this capsule around the cell. So the donor cell here has this capsule, whereas this cell doesn't. Okay, but it's passed the gene in this plasmid to the neighbouring cell, which has then created this capsule, and as you can see, this bacterial cell can now produce this capsule because the gene for making that capsule is on that plasmid. Okay? Now, why do bacteria do this? The answer is that it helps their survival, in that a useful gene, like one bacteria, which could confer some form of survival on the bacteria, can be passed to neighbouring cells within a colony to confer whatever that survival characteristic onto neighbouring cells, okay, which enables bacteria to adapt very, very quickly to changing environments, okay, particularly appropriate these days in the terms of MRSA and C. difficile, which are bacteria which are very highly resistant to any form of antibiotic that we give them. Okay, so working that through, okay, imagine, for example, that this cell here is resistant to a particular antibiotic, okay, perhaps due to the fact that it produces this capsule which protects it from an antibiotic, and this cell isn't. Okay, now put in this antibiotic, what will happen very quickly is that this cell here will share the gene with neighbouring cells to protect it from the antibiotic, okay, i.e. growing a capsule around it. So suddenly this cell here is also resistant to our antibiotic, okay. This cell reproduce and produce lots and lots of other cells, all of which are resistant to our antibiotic, and we very, very quickly get a new strain of bacteria which is antibiotic resistant. Okay, so that explains why plasmids are so useful to bacteria, but they're also been a very, very useful tool for genetic engineers to get DNA from one cell into another. Moving on to the next slide, which is quite complicated, is it looks at the idea of genetic markers, another tool that genetic engineers have to use. All the things we've talked about plasmids and getting genes into plasmids and using enzymes, restriction enzymes and DNA ligase, and then being able to transform bacteria using techniques to get this engineered plasmid into cells. Obviously we can't see what's going on, it all happens in a test tube, it's all very abstract, and it's actually quite a hit and miss affair. So what we need to be able to do at the end of all these experiences, work out which bacteria have taken up our genetically engineered plasmid and which bacteria haven't. Okay, and the way that we do this is we put things called genetic markers into our plasmid which enable us to do this. And quite often what we do is we also engineer into a plasmid some form of gene which confers resistance to an antibiotic, okay, and we'll see why that's important in a second. Okay, so I've said, looking at your notes, when introducing recombinant plasmids to the bacteria, there's no way of discriminating between transformed bacteria, i.e. the bacteria that have taken up the plasmid, and bacteria that are haven't been transformed, i.e. bacteria that haven't taken up the plasmid. Okay, to overcome this problem, plasmids have marker genes or gene markers inserted into them, as well as the target gene. Okay, the marker gene is often a gene that confers antibiotic resistance. Okay, the idea being that by then exposing our bacteria to the antibiotic will kill all the bacteria which haven't been transformed. Okay, because they haven't taken up the plasmid. They won't have taken up the ability to be antibiotic resistant, and they will therefore be killed. Okay, so bacteria can be exposed to antibiotic, and only transformed bacteria survive. So that's the overall principle. It's slightly more complicated than that, and I'll try to explain that through the following diagrams. Okay, so what we actually often do is we engineer a plasmid to have two genes, okay, which are resistant to two different antibiotics. Okay, so here we have a plasmid we've engineered. Okay, and this plasmid has a gene which confers resistance to an antibiotic called tetracycline. Okay, and it also has a gene which confers resistance to another antibiotic called ampicillin. Okay, one of the original penicillin type antibiotics. Okay, so what we do is that the idea is that we have a restriction enzyme which actually forms a staggered cut in the middle of the gene for tetracycline resistance. Okay, and the idea is that we then insert the gene in the middle of this tetracycline gene. Okay, that might seem a bit odd, but I'll explain why we do that. Now, as I said at the start, it's a fairly hit and miss affair of what the plasmid might do. When we add the ligase, okay, the ligase can do one of three things. Okay, it could just knit our plasmid back together again. Okay, so it could take this plasmid and just knit it back together again, so we end up with what we started with. Okay, it could do what we want it to do and knit our insulin gene, okay, or our target gene, into the middle of the tetracycline resistant gene. Okay, or it could just take the gene and convert it into its own little loop. Okay, because it could take the end of this and stick it to the end of that, because these would have complementary sticky ends, so we could end up with our loop. Okay, so we have option A, option B, option C. Okay, so what we would then do is that we would, if you look over here, what we would then do is spread our bacteria. Okay, so we've tried to get these plasmids taken up by bacteria, okay, by heat shock treatment or electroporation or whatever, and we then plate our bacteria onto an agar plate, petri dish with agar into it, and allow the bacteria to grow. And what we would find is that we would have different types of bacteria growing on this plate. Okay, we could have bacteria which has plasmid A, so the blue ones here have this plasmid, okay, and would be resistant to both ampicillin and tetracycline. Okay, we'd have ones which, the green ones, which are B, which would have our target plasmid. Okay, that's what we want. We want plasmids like this in our bacteria. So we've got the green ones are the ones we want. Okay, or we could have bacteria with just the gene for insulin in it. Okay, we don't really want that. Okay, so we'll be able to get rid of that, or we might have just other bacteria which might be living on the plate. Okay, so what we then do, okay, so we've grown our plate like that, and then there's another trick that microbiologists use called replica plating. Okay, so what they do is they take a wooden block which has got a piece of velvet attached to it, and they blot this wooden block with the velvet onto the plate. Okay, and the bacteria stick to the velvet. Then they transfer the velvet block to a fresh agar plate. Okay, but this agar plate, okay, so we've transferred by using replica plating, we transfer this to this plate here, but this plate here has embedded into the agar the antibiotic ampicillin. Okay, and what the ampicillin will do is the ampicillin will kill all of the bacteria that don't have the ampicillin resistant gene. Okay, so if the bacteria's got one of these in it, okay, ampicillin resistant here, it will survive, and similarly if it's type B, it will also survive. Okay, so as you can see here, the green ones, okay, the type B, they survive, and the A ones here, those survive. Okay, and all the other ones, okay, the ones with that in it will die, okay, because there's no resistance to ampicillin, and any other bacteria that might be in the plate will die also. Okay, the problem here though is we don't know which bacteria have got plasmid A and which have got plasmid B. So what we then do is we then, by replica plating, we then transfer this to this plate here, okay, which has got tetracycline in it, and the tetracycline ones will kill off the target ones, which are the B's, okay. So the only ones that are left here are the ones with A, okay. So what does that tell us? Okay, we can go back to this plate here to tell us that that colony, and that colony, okay, contain the target bacteria, i.e. the bacteria which have got this plasmid in it, okay. So we know that these are bacteria which have been transformed, or bacteria that have been transformed with the plasmid that we're trying to get into that bacteria. Okay, so we can then take these and then we can put those into, let's say, a fermenter, and those multiply and will produce our target protein, which in this case was insulin. So that concludes the second of our genetic engineering screencasts.